Thanks very much for joining us and so early on California time. I've asked you to just remind us who you are. I know you know many of us, but for those who didn't overlap with you, we're really excited to see your work and to interact with you over the day. So thanks very much for joining us.
Yeah, thank you for inviting me. I'm excited to get to talk to this group and to get to meet with some people afterwards. So my name is Burcu Darst, and I'm currently a postdoc at USC. But I previously was a graduate student at the University of Wisconsin, and I worked with Corinne Engelman on genetics and metabolomic risk factors of Alzheimer's disease and Alzheimer's risk factors, doing a lot of like integrative multi-omic type of work and some work while I was with Corinne on polygenic risk scores. So the work I'll present today, and I graduated in 2018, so I've been here for about 2.5 years now. So the work that I'll present today is really focused on polygenic risk scores, and in particular, developing them and applying them in diverse populations. And I'll primarily talk about prostate cancer. That's where my main area of research is right now. And then in addition to actually like developing the polygenic risk score and applying it, I'll talk about some subsequent work that we've done to better understand genetic risk of prostate cancer using the polygenic risk score by incorporating whole exome sequencing data to investigate rare variants with polygenic risk scores. And then also looking at metabolomics to try to understand what it means to have high genetic risk of prostate cancer. And then the last thing I'll talk about is a very similar application of developing this multi-ancestry polygenic risk score, but applying it to a type 2 diabetes multi-ancestry population. And if you have any questions throughout, you're certainly welcome to stop me at any point. Okay, so I'll go ahead and get started. So just to give a little bit of background, polygenic scores quantify genetic predisposition to a heritable trait, and it's calculated typically as a sum of risk alleles weighted by corresponding variant-specific effect sizes. This paper by Torquemani et al. Does a really nice job of describing the potential clinical utility of polygenic scores. And in particular, they categorize it into 3 different areas. The first is that they could be used to inform disease screening, such as whether or not someone should be screened for a disease, at what age, and how frequently they should be, if they should be screened for it. They could also be used to inform whether somebody should receive therapeutic interventions for disease treatment. And they can also be used to inform life planning. And this is really more the personal clinical utility of genetic scores, such as something that can inform your financial and life planning. And this paper also gives a really nice figure here where they kind of try to graphically display what the clinical utility of genetic risk scores could be, where we add genetic risk to clinical risk to get a more comprehensive measurement of risk. And in particular, if you have a person, such as this bar here, who has clinical risk that's really borderline, but you don't yet know what their genetic risk is, you can incorporate their genetic risk to get a better measurement and potentially recategorize them with a little bit more confidence if they're potentially low risk or if they do meet that threshold for clinical action. So this is a really nice paper, and I've listed some references at the bottom of some of the slides I'm showing if you're interested to learn more about them. So the problem with polygenic risk scores is that they're typically based on GWAS studies, results from large-scale GWAS studies. However, GWAS studies tend to be based on predominantly European ancestry populations. So this paper from Alicia Martin et al. From 2019 shows that between the years 2006 and 2018, the overwhelming majority of participants included in GWAS studies are of European ancestry, shown by the red in this figure. However, this is not at all reflective of the distribution, the global distribution of individuals, where we see only about 15% of the global population consists of European ancestry individuals. This paper goes on to show that when we develop genetic risk scores or polygenic models of various traits, that the predictive accuracy, which is shown on the y-axis here, is increasingly poorer as populations become increasingly divergent from European ancestry. So in particular, the predictive accuracy in African ancestry populations is only about 25% that in European ancestry populations. And you can imagine that this could really exacerbate existing health disparities, especially for a disease where the incidence and mortality rates are much higher in a particular non-European ancestry population. So prostate cancer is an example of one of these diseases where we see that African ancestry men have incidence rates that are substantially higher than all other populations that are looked at in this figure, and mortality rates, again, that are much higher than other populations. Another way we can look at this is using rate ratio. So now we're showing rates in each population relative to rates in European ancestry men, which is represented by the black horizontal bar. And you can see now that African ancestry men have incidence rates that are about 1.5 to 1.75 times higher than they are in European ancestry men between the years 2000 and 2017. And we see an even bigger disparity when we look at mortality rates, where mortality rates are about 2 to 2.5 times higher in African ancestry men than European ancestry men. And this has been true for the past couple decades. So taking this into consideration, again, with this evidence that polygenic scores don't typically do as well in African ancestry individuals, you can imagine how this could potentially exacerbate health disparities. And just to give you an example, there is currently a clinically available polygenic risk score for prostate cancer provided by Ambry Genetics. However, it's only offered to men who are of Northern European ancestry because the variants that they include were identified in European ancestry population, and the weights that they use are European ancestry weights, so it wouldn't do as well in non-European ancestry men. So to give you a little bit more background on prostate cancer in 2021 is estimated to still be the most commonly diagnosed cancer in men in the US. And it's the second leading cause of cancer death of men in the US. Despite how common it is, there are very few established risk factors. Some of those include age, with older age being associated with increased risk of disease, ancestry, as we've just shown, and family history, with the heritability being about 58%. And this is a really important note because prostate cancer is one of the most heritable cancers. And I think because of that, the results that I'm going to show today look different than they might in other traits. And genetic studies to date have shown this to be true, that it is a really strongly genetic disease. So this review was published in 2018, and they showed that at the time, there were about 180 genetic variants that were associated with prostate cancer identified in GWAS studies. And in aggregate, these 180 variants account for about 28% of the familial relative risk for prostate cancer in European ancestry men. And we also recently showed that these 180 variants account for about 28% of the familial relative risk in African ancestry men also. However, these 180 variants were identified in predominantly European ancestry men and populations, and so the variants and the weights are European ancestry and may not work as well in a non-European ancestry population. So the work that I'm going to show today is really trying to include as many non-European ancestry men as were available at the time that we did this study to really try to get a better understanding of genetic risk of prostate cancer across diverse populations. So we did this GWAS meta-analysis, and this is the largest and most diverse prostate cancer GWAS meta-analysis that's been performed to date. We had a total of 230,000 men across African, East Asian, European, and Hispanic populations with the sample sizes shown in this table. We analyzed all of these different GWAS studies using fixed effects, inverse variance weighted meta-analyses performed in metal. And we did this first within each population, and the results are shown here. And then we did it across all populations to get multi-ancestry results with the results shown in this figure here. Once we had these results across populations and within populations, we did fine mapping of any significant regions, either within a population or in the multi-ancestry GWAS. And we did this fine mapping with a Ford stepwise selection using 800 kb window, starting with the smallest p-value and adding ranked by p-values. We did this within each population to get population-specific conditional summary stats, and then we meta-analyzed those population-specific summary statistics using a fixed effects meta-analysis, again, to obtain multi-ancestry conditional summary statistics. In total, we identified 86 novel risk variants. For 62 known risk variants, we were able to identify stronger markers of risk using fine mapping. And for 121 variants that were known, they remained the lead signal in those regions. So in total, we the number of known prostate cancer variants is 269. And again, this is not just in the multi-ancestry, but it's also within populations. If a variant was only significant within a population, we include that in the number in the 269. So when we take these 269 and look at the familial relative risk, we see that in aggregate, these account for about 34% of the familial relative risk in East Asian men and up to 43% in European and African ancestry men. And just as a reminder, the previously known 180 European ancestry variants accounted for about 28% of familial relative risk. So these additional variants plus the fine mapping plus the multi-ancestry weighting is really showing a pretty substantial improvement in what we can explain for familial relative risk. So we took these 269 variants and developed a polygenic risk score, which is calculated as a weighted sum of the number of risk alleles carried by each participant, where the number of risk alleles is represented by G for a given individual I and for a given variant M. And the weight that we use is beta, and that beta is a multi-ancestry conditional weight for each variant. And for this, we actually use, instead of the inverse variance weighted fixed effect meta-analysis, we use inverse z-score weighted fixed effect meta-analysis. And this is just to upweight any variants that are really significant in one population and don't have as much evidence in other populations. So once we have the PRS calculated in all participants, we can plot that PRS distribution in cases shown by the solid line and controls shown by the dotted line. And then we like to stratify our participants into deciles using cutoffs determined in controls as shown in this kind of toy figure here. And so we typically like to compare the number of cases and controls that fall into, for example, the top decile to the number of cases and controls in the reference average PRS category, the 40 to 60% PRS category. And this is just showing one example or one population, but we do this within each population separately and determine these cutoffs separately within each population. So once we have here, yeah.
I'd call it an architecture question. Are there big hits in prostate cancer? Are there like ApoE type variants, or is there? Yeah, can you say a little bit about that?
There are, and some of them are actually population specific. So one of the big hits is the HOXP13 gene has a particular variant that has a really large effect. I think the odds ratio is something around 4, and it's just one variant, but it's pretty rare. The minor low frequency or the risk low frequency is usually less than 1%, but it's fairly common in cases, obviously. And that's only present in European ancestry individuals. And then in African ancestry men, there's a variant in the 8q24 region, and I'll talk a little bit more about that later today, just the 8q24 region, not this particular variant, but this particular variant is only present in African ancestry men, and it has an odds ratio of about 2 just for one risk allele. And that's a pretty substantial effect, and that variant alone accounts for like 9% of the familial relative risk in African ancestry men. So it really kind of drives a lot of the risk that we see in African ancestry men. And that variant is also really promising because usually, I'll talk about this, well, I might not mention this actually, but usually when we look at common variants, they're associated with prostate cancer overall risk, but not aggressive disease specifically. But this 8q24 variant in African ancestry men actually is also associated, it's more common in men with aggressive disease. So that's kind of an interesting finding as well.
Is there any story about the presence of these big hits in human populations, like what the potential upsides of carrying these are?
Well, there are theories, but I don't know if any of them are proven. But one of the theories is that maybe, like, why do men have high genetic risk at all? But it's a late onset disease. So maybe there's something about fertility in earlier age, but I don't think there's anything that's been proven yet. But that's one theory, that maybe having high genetic risk for prostate cancer has something to do with increased fertility.
Thanks, that's helpful.
Yeah, no problem. Okay, so we calculated the polygenic risk score across all participants in our GWAS study that we had the individual level data available for, and those sample sizes are shown in this table here. And then the primary results for this analysis are shown in this figure here. So just looking at the top PRS decile, the 90 to 100% category, we see that the odds of prostate cancer is about 5.1 in European ancestry men, and that's compared to European ancestry men in the average 40 to 60% category. It's a little bit lower in East Asian men, a little lower in Hispanic men at 4.2, and lowest in African ancestry men where the odds is 3.7. And we replicate these findings in an independent sample of about 2,600 African ancestry men from the California-Uganda study, and 200,000 European ancestry men from the UK Biobank. And here we see pretty similar odds ratios as what we observed before. And I really want to emphasize that this difference between 3.5 and 4.2 is actually really promising, because we typically see in polygenic risk scores much bigger differences between populations. But in prostate cancer, we actually see that this polygenic risk score, which it is a multi-ancestry polygenic risk score, but it seems to be fairly transferable across populations, which is really promising and also really unusual for a polygenic risk score. So we also look at area under the curve to evaluate the discriminative ability of the prostate cancer PRS. Looking at our European ancestry replication sample, we see that in a base model with just age, the AUC is about 0.78. We see very little improvement when we add family history. We see pretty big improvement when we add the PRS to the age-only model. And again, when we add family history to the age plus PRS model, we see really little improvement. So this is suggesting that the PRS is a better measure of prostate cancer risk than family history. And this is consistent with what previous studies have shown. In our African ancestry replication sample, we unfortunately didn't have family history available, so we weren't able to evaluate that. But in our age-only model, we see that the AUC is about 0.53, much lower than what we saw in the European ancestry model. And then when we add the PRS, we see a really substantial improvement up to 0.68. And this is a much bigger improvement in African ancestry men than we saw in the European ancestry sample. Do you have a question?
Hi, Burcu. Yeah, sorry, this is great. I have a question. I'm trying to wrap my head around there's so much diversity, genetic diversity in Africa. Mm-hmm. How did you guys go about selecting? Are these all African Americans? Are they people that come from whose ancestors come from different regions of Africa? Or did you target a certain region like West Africa because they've seen higher rates of prostate cancer in certain areas of the West African genome? Or can you talk a little bit about that, and whether the prediction is different across different regions of Africa? Or thank you.
That's a good question. So really, like for a GWAS, we've been finding that the most important, the biggest way we can improve our ability to identify variants and predictive ability is just to have the largest sample size possible. So we include any men with African ancestry that we can. So this does include a large number of African American men, but it also includes some men from Ghana, men from Uganda, men from the Caribbean, the French West Indies. So really like anyone that we can, but there is this big question of heterogeneity, because there certainly is a lot of heterogeneity within African ancestry populations. And I have a slide that I can maybe show later that kind of touches on that. Also, if there's time at the end, I could maybe show this. I think it's a really interesting thing to look at, but there certainly is there are a lot of differences within these different African ancestry populations.
Great, thank you. That's interesting.
This is Yuchang. I have another question. So this cancer is very heritable. I wonder why when you add family history in the model, the AUC did not increase. It increases only a little bit.
Yeah, I mean, I think the reason is probably because the polygenic risk score is capturing a lot of what the family history variable captures. I think there's just a lot of overlap there in those two. And also, I think the polygenic risk score is probably might be like in terms of trying to capture family history is kind of a muddy variable, because we're totally relying on people to accurately report their family history, which is surprisingly really hard to do, I've heard. So I think family history is not always very reliable. It really depends on how the question's asked and how it's collected. So I think that's probably part of why the PRS does better than family history.
Okay, that makes sense. Thanks.
Yeah. Is there another question?
Yeah. Hello, everyone. I've been wanting to join in with these meetings since getting to Wisconsin, but it's just been crazy. I'm really excited about your talk. This is all very fascinating stuff. I know enough about population genetics and social genomics to be dangerous. But here's just the question. So my understanding is that the chips that we use to encode genomic data for African-based populations are different than the European-based. So I'm wondering how you encoded your data. And I'm wondering, how do you reconcile the need for the difference between the greater diversity within sort of African descendant populations with the overall smaller sample size that you end up getting? Just your thoughts on it.
Yeah, that's a good question. So for the first one, there are some more recent genotyping arrays that are really targeted at trying to incorporate genetic variation from more diverse populations. This certainly is a problem because I think most chips that have been used until very recently were designed with around the LD structure of European ancestry populations. So the variants that were captured were not representative of anyone non-European. And I think that's been problematic. But now we have like the mega array and the H3 Africa array, which are really targeted at more diverse populations. So they try to incorporate variation that is common in those populations, hoping that we can better capture the genome. And then of course, the thing that we do once we have these genotyping chips is that we impute them up to whole genome data. And that imputation is based on diverse populations, like the 1,000 genomes it has this whole genome sequencing that we always use, almost always use for our imputation. There are like really popular imputation panels that are European ancestry, like HRC. But for our studies, we've been using 1,000 genomes for imputation because that's more reflective of diverse populations. And now with TopMed that's available, we'll be able to hopefully even do a better job with imputation, capturing more diverse populations. So that's how we've addressed that so far. But that is potentially a problem. I think a lot of I'm not sure exactly what chips we used across these populations, but it is possible that at the time the study was done, it might not have been the most optimal chip to capture such variation in some of these populations. And then, yes, in terms of the heterogeneity, so I think I do think that it influences results. And I think there are probably better ways we can do our GWAS analyses, like actually doing the association tests to utilize that heterogeneity. But I think the way that we typically do it, that heterogeneity might be lowering our power to detect associations. Because really for a GWAS study, having a homogeneous population increases your power because, certain variants are going to be more common than others. And but yeah, that is a problem. And I think there are probably better ways that we can do GWAS studies in the future that maybe incorporate mixed models to really utilize the heterogeneity across and within populations instead of being kind of underpowered because of it.
Yeah, no, I really appreciate your attention to the diversity of the samples here. And so I think, as far as I can tell, you're probably doing best practices. Last question is why we do have other populations in the sample. And so why the focus strictly on African as opposed to European, when in fact, one might argue that the sort of Asian populations had even a lower baseline and might be the appropriate, that is to say that Europeans also have elevated risk relative to the Asian populations. And so how are they the sort of norm or baseline in this study?
Yeah, so we don't really have, so the reason that I'm showing African ancestry and European ancestry replication is only because those are the only studies that we were able to get additional GWAS data that weren't already included in our GWAS. We couldn't find any additional European or East Asian ancestry samples to be able to do replication. It was just a limitation. But that now, since we did this study, we've been collaborating with MVP, the Women Veterans Program, and they have a pretty large number of men, first off, because it's MVP, And they have good diversity, so we've been able to do more in there. But actually, even in that study, they have a pretty small number of East Asian cases, prostate cancer cases, so we're still somewhat limited in our ability to replicate. But I think it's Riken. I'm not sure exactly which study it is, but there is another prostate cancer East Asian GWAS analysis that they recently got a larger sample size. So we might be able to use them for additional replication in the future, but this was just a limitation. There was otherwise we would certainly incorporate as many others as we could for replication, but it was just a limitation at the time that we did this. Yeah. Okay, so there's a few additional things we did with the polygenic risk score in this study. The first was to stratify by age and reevaluate the odds ratio. So here we're looking at the odds ratios in men who are less than or equal to 55, and odds ratios in men who are greater than or equal to greater than 55. And you can see that the odds of prostate cancer is much larger in men who are younger than men who are older. And this was true in our discovery sample, shown by the bright green, and our replication, shown by the bright darker green. And this is looking in our European ancestry sample. But when we look in our African ancestry sample, we see similar effects, although the differences are not quite as strong, but we do typically see that the risk of prostate cancer is greater in younger men than it is in older men. And this makes sense. This is suggesting that the PRS would most likely be most informative of prostate cancer risk in men who are younger, which makes sense because the older a man is, the more time he has to accumulate environmental risk factors that could be contributing to his prostate cancer risk, whereas younger men are more likely to have a stronger genetic component. And just to reiterate, I do focus a lot in some of these subsequent analyses I'm showing on African and European ancestry. And that is, again, just a limitation of sample size, because when we didn't have a big enough sample size to be able to stratify some of the other populations. So we also look to see if the polygenic risk score is associated with age at diagnosis for prostate cancer. So just focusing on one color at a time, which is one population, looking at the East Asian men, for example, we see that on the x-axis, this is the different PRS categories, and the y-axis is the mean age at diagnosis. So East Asian men with low PRS have an age at diagnosis that's close to 71. But then if we go down to the higher PRS, those in the highest percentile have an age at diagnosis that's closer to 67 or 68. And you can see this downward trend of having a younger age at diagnosis is true across the different populations that we look at, suggesting that higher genetic risk is associated with a younger age at diagnosis, which is often associated with more aggressive disease. But the thing I want to point out is that it looks like there are big differences at age at diagnosis between populations, but this is really just a sampling, differences in sampling strategies. So we aren't really able to evaluate whether there are true differences in age at diagnosis between the different populations. So this is really just intended to look within a population to see that there's a younger age at diagnosis with higher genetic risk. So one of the most important questions to address for prostate cancer is, can the polygenic risk score distinguish between aggressive and non-aggressive disease? And we unfortunately saw that the PRS is essentially about the same between aggressive and non-aggressive cases. So I'm not showing those results here, but what I am showing is that we look to see how aggressive cases are distributed by the PRS. And what we see in particular, looking at European ancestry men first, is we see about 50% of aggressive cases represented by the bright green fall into the top 80 to 100% PRS category. Likewise, in African ancestry men, we see about 45% of aggressive cases are in the top PRS quintile. So this is suggesting that the PRS would likely be able to capture a large proportion of aggressive cases if it was used clinically, which could have important implications. We also look at absolute risk of prostate cancer by age, which is on the x-axis here, and PRS category, which is shown by the different colors here. And you can see that as the PRS increases, there is much higher absolute risk. And we can kind of look at this black line here as the average population risk. So in European ancestry men, lifetime risk of prostate cancer is about 10%. But you can see if we just focus on the top PRS decile, men in the top decile have a lifetime risk of about 38%. And we see 38% also in African ancestry men. It's a little bit lower, 26% in East Asian men. and 31% in Hispanic men. We also look at absolute risk incorporating family history, and now you see a similar picture, but we additionally see that adding family history really shows a pretty big increase in risk on top of the polygenic risk score. So this is suggesting that the use of both the polygenic risk score and family history would likely be more informative than just looking at one or the other. So up to this point, we've been talking about a genome-wide significant PRS, meaning that we only look at the 269 variants that are significantly associated with prostate cancer. But in a lot of studies, a lot of recent studies have been showing that using a genome-wide PRS, using variants that go below the significance threshold, could actually improve the predictive ability for a number of different traits. So we wanted to see if this would be true for prostate cancer. So we developed a genome-wide PRS just using this p-value cutoff of less than 10 to the negative 5, including any variants up to that threshold. And we evaluated it in an iterative fashion. So we first started off with a polygenic risk score that has the most significant variants in it, which is shown by the left side of this on this graph. And then we add one at a time, one more variant to the model ranked by its p-value. So that's shown on the x-axis here, the number of variants that's included in any given model. And for each of these different PRS models, we evaluate the AUC, which is the black line that corresponds to the left y-axis. And we also evaluate the odds ratio for the top PRS decile, which is the blue line that corresponds to the right y-axis. So just focusing first on the first 50 or so variants that get included in a PRS model, and these are the most significantly associated with prostate cancer, this is where we see the most dramatic improvement in both AUC and odds ratio. But we see adding additional variants beyond those 50 up to our 269 PRS, we do see continued improvement in terms of AUC and odds ratio. But beyond that, we see that the AUC really tapers off, as does the odds ratio, and we see that it even declines a little bit at some point. So this was looking at our European ancestry replication sample. But when we look at our African ancestry replication sample, we see a pretty similar picture where the first 50 or so variants have the most dramatic improvement in terms of AUC and odds ratio. We see continued improvement up to our 269. And then we actually see that adding additional variants that are not genome-wide significant just adds noise to this model. So these last two figures that I showed, we're focusing on the multi-ancestry p-value and the multi-ancestry weight. But we wanted to see if using maybe African ancestry p-values and African ancestry weights could improve what we see in our African ancestry sample. So we tried that, but as you can see, it didn't look quite as promising. We do see, again, this really rapid improvement with the first 50 variants and up to the 269 PRS, but adding variants beyond the 269 actually just shows really, really poor improvement. So our multi-ancestry PRS seems to be doing a lot better. And the other thing I want to point out is that it looks like there's a lot more variation in the African ancestry genome-wide PRS that I showed, but that's just because of the much smaller sample size here than we had in the UK Biobank. So this is suggesting that a genome-wide PRS may not improve predictive ability of prostate cancer compared to a genome-wide significant PRS, but there are many other ways that a genome-wide PRS can be evaluated. And this is something I'm currently working on, so I'll show some very preliminary results for that. But first, before I show that, I just want to kind of address this question of, so we've been using these multi-ancestry weights, but could using population-specific weights work better? So here I'm focusing on just the African ancestry population because that's where we see that we really need the most improvement in our PRS. So to see if using African ancestry weights could improve the PRS further. And we actually see that the results are very similar. So these multi-ancestry weights on the left are what I showed graphically earlier. And on the right, we're looking at African ancestry weights, but we see that the numbers are actually pretty similar. It doesn't really change too much. And I think part of this is probably a sample size. I mean, I think it's impressive that we do see pretty similar odds ratios here because our African ancestry sample was only about 20,000, whereas the full multi-ancestry sample is about 230,000. But I think in the future, when we have larger sample sizes of non-European ancestry men, we can probably do much better than what we're doing right now. And I think this is supported by this paper that was recently published by Taylor Cavazos and John Whitty. And the approach that we're using is kind of represented by the right side of this figure, where you take variants that are significant in fixed effects meta-analyses across populations. And So maybe to give a little bit more background, so this is based on simulated data. They simulated an equal number of European ancestry individuals and African ancestry individuals. They do a GWAS within the European ancestry to identify European ancestry variants. Then they do a GWAS within African ancestry to identify African ancestry variants for a simulated trait. And then they do a fixed effects meta, combining the two studies to identify variants that are significant across studies. So what we're showing is kind of represented by here. They're showing the, maybe this bar here we'll look at first. So they develop a PRS that's based on variants that are identified in the multi-ancestry sample. They apply a multi-ancestry weight shown by purple. They also try applying African ancestry weight shown by pink, and they're testing it now in an African ancestry sample. And you can see that the weights do pretty similar. The multi-ancestry and the African ancestry weights do similar, which is kind of what we're seeing here. But they also show, if we look on this, where we use the African ancestry SNPs instead, if they test in these African ancestry SNPs in an African ancestry population, using either the multi-ancestry weight shown by purple or the African ancestry weight shown in pink, it does much better than the fixed effect analysis does. I think what we're doing is a little bit better than what's represented by this fixed effects meta-analysis, because we also do fine mapping and we also take variants that are significant within specific populations. So not just multi-ancestry significant, but this is really showing that if we had larger samples of non-European ancestry individuals, we would likely be able to build much better predictive models across populations. So I think in the future, as we start to focus more on incorporating non-ancestry individuals, we'll probably be able to do much better in our polygenic risk models. So, okay, so to show some of the genome-wide results. So for this, we use the 1.2 million variants that are from HapMap, which are recommended for these different, I'll show 3 different genome-wide approaches. And we take the weights, the multi-ancestry weights and the African ancestry weights, and we calculate new weights using LD-PRED2. And you can see this is showing the results from that. The results we get are actually pretty similar to what we see here, maybe with a little bit lower odds ratios in the top decile. And then when we try the African ancestry weight, again, we see pretty similar results. It doesn't really show that this is improving the predictive ability to use this genome-wide approach. We also try using PRS-CSX, which is all of the 3 genome-wide approaches that I'll show are all Bayesian methods. But this one is a recent method that was just posted on medRxiv recently, where it really tries to, it utilizes multi-ancestry GWAS summary statistics, whereas the other approaches that I'll show just look at one set of summary statistics at a time. But this one looks across different populations. But even with this one, we still see that the odds ratios are actually pretty similar to what we see, actually a little bit lower than what we see in our using our genome-wide significant PRS. And that was true for both the multi-ancestry weight and the African ancestry weight. And then the last approach we look at is this EV-PRS. This is an empirical Bayes method that actually doesn't use an LD reference panel. The other 2 did use an LD reference panel to try to reweight variants. But this one just uses summary stats. And for this, we again see that it doesn't really show improved odds ratios compared to what we saw using our 269. Just to kind of summarize these different methods, looking at the AUCs, you can see that our genome-wide PRS has an AUC of about 0.68, using both the multi-ancestry and the African ancestry weight. And the other approaches that we look at have lower AUCs pretty consistently. So it does seem like our genome-wide significant PRS is doing really well, maybe better than a genome-wide model would be able to do. But again, I think this is something that's very specific to prostate cancer and certainly would not be true for other traits. And this question of genome-wide PRS is important to address across traits because it may often be the better approach. There are other approaches that can be used for genome-wide PRS, and I'm listing some of those here. And these are things that we're still exploring, but these 2 really kind of incorporate fine mapping a little bit more, so they could be a better approach than the ones that we've shown. So to kind of summarize the progress that we've seen in prostate cancer. So with this study, we added about 20,000 cases. Most of them were of non-European ancestry. And these 20,000 additional non-European ancestry cases that were added to the study really lead to a pretty substantial improvement in odds ratios. So with the previously known 180 variants, looking, for example, in African ancestry men, the odds of prostate cancer in the top PRS decile was about 2.5. But with our newer 269 variants, it increases to about 3.5. And we see this improvement across all populations. I think this really emphasizes that incorporating non-European ancestry individuals is it's a win-win for everybody. It's something that we really need to do, and it'll show, I think it will just really help us understand the biology and improve predictive models across populations. And for us to see further improvement in these PRS, it will be really important to increase our number of non-European ancestry men that are included in these studies. So one of the next important steps of this work is, can it improve, can it actually be used to improve our ability to identify aggressive disease in a clinical setting? So we've tried to start to address this question using participants from the multi-ethnic cohort. And here we had a pretty diverse sample of Japanese, African-American, Latinx, Hispanic, European, and Native Hawaiian individuals. And we can see that compared to PSA, which is what's typically used in a clinical setting. So we have this panel of different PSA markers, which are captured by the 4K panel, and then looking at other PSA markers as well. But looking just at these PSA markers, you can see that when we add the PRS represented by the gold, we consistently see across populations that there is at least a slight improvement in AUC across all populations. And in particular, we see the largest improvement in African ancestry men. And this is true in both aggressive prostate cancer and when we look at death due to prostate cancer as the outcome. And an important thing to note here is that the PRS used in this model was based on the previously known 180, which are predominantly European. So looking at our 269 multi-ancestry will likely show even better improvements in discriminative ability. But there are many additional ways that this will need to be evaluated in the future to make sure that this could actually be used to reduce health disparities instead of increase them. So another thing, a next step that we wanted to look at in this study, another study, was to see if the effect of rare pathogenic variants on prostate cancer risk is modified by polygenic risk. So a lot of studies have recently shown this to be true in other traits. And we wanted to see if this is true in prostate cancer too. So we focused just on carrier status for pathogenic, likely pathogenic, and deleterious variants in these really well-established prostate cancer risk genes, such as HOXP13, BRCA2, ATM, and CHEK2. And we used data from the UK Biobank, where they recently released 200,000 whole exome sequences, which included about 3,500 cases and 78,000 or 77,000 controls. So we look here, where I'm showing the odds ratio, the odds of prostate cancer in carriers for these pathogenic variants, and in non-carriers, shown by the dark blue. And we're showing this across different PRS categories. And in particular, you can see that there does appear to be some effect modification where carriers who have low polygenic risk have very low odds of prostate cancer. It's essentially 1, whereas carriers with high polygenic risk have an odds of prostate cancer that's close to 11. Likewise, in non-carriers, we see this protective effect for men with low PRS, but in non-carriers with high PRS, we see an odds of prostate cancer of about 5. When we look at the absolute risk of prostate cancer by age 85 in carriers, it's about 26%, but in non-carriers, it's about 11%. However, when we actually incorporate polygenic risk information into these measurements of absolute risk for carriers and non-carriers, we can actually see that there's a pretty wide range in absolute risks. In particular, when we look at non-carriers with absolute risks in this figure are represented by the solid line for non-carriers, and carriers are represented by the dotted line. And the different PRS categories are shown by different colors. So looking at non-carriers with low PRS, which is this line right here, you can see that the absolute risk of prostate cancer is about maybe 2% by age 85. But in non-carriers who have high PRS, that absolute risk, which is the red solid line here, absolute risk reaches about 30%. Likewise, in carriers with low PRS, which is the dotted gray line, they have an absolute risk of about 10% by age 85. And in carriers with high PRS, the absolute risk is about 55% by age 85. So this is really clearly showing that considering rare variants in conjunction with PRS will likely be able to show pretty great improvements in predictive models. Another interesting finding from the study was that we saw that, I mentioned this HOXP13 G84E variant earlier. This is a really strong risk factor for prostate cancer. If we look at the carrier frequency of this variant stratified by PRS categories, you can see that in cases, which is the gold bar, they have the highest carrier frequency of this variant, where it's about 4.5% carrier frequency, whereas in men with high PRS, that carrier frequency is about 0.5%. And then likewise, when we look at the average PRS by carrier and non-carrier status, you can see that carriers have on average a lower PRS than non-carriers. So this is really suggesting that there could potentially be two different pathways to developing prostate cancer. You either have a high PRS and that contributed to your prostate cancer, or you're a carrier for these rare pathogenic variants, and that maybe contributed more heavily to your prostate cancer status. However, these are not mutually exclusive things, and having both of them could likely would really substantially increase your risk of prostate cancer. But this is also suggesting that sequencing men with low PRS could be a good way, a powerful way to identify additional high penetrant risk variants that are associated with prostate cancer. And similar studies are needed in African ancestry men, and this is actually something that we're currently working on. Since I'm kind of running out of time, I might skip over. The next thing I was going to talk about is metabolomics, because I know that there are a lot of people interested in that, but maybe if there are questions, I can talk about it. But I'll skip to the last part of this talk, which is to talk about the type 2 diabetes PRS work that we've done, and I'll just talk about that really briefly. So type 2 diabetes is another disease where we see these health disparities between populations. In particular, looking at these incidence rates here across populations, you can see that African ancestry, Asian ancestry, and Hispanic individuals have higher incidence rates for type 2 diabetes than European ancestry individuals. However, type 2 diabetes does have an environmental component, which we can see when we look at these risks by different education strata. And you can see that in particular, individuals with lower education are more likely to have type 2 diabetes, which indicates that there is an environmental component to this disease. And then when we look at prevalence, we similarly see these disparities where individuals who are of Amerindian or Alaska Native ancestry have the highest prevalence of type 2 diabetes, followed by Hispanic individuals, African ancestry individuals, Asian, and then with European having the lowest prevalence. And type 2 diabetes does have a heritable component, but heritability estimates have a really wide range for type 2 diabetes, from about 20 to 80%. So we did a multi-ancestry Gwaspin analysis of type 2 diabetes, very similar to how we did it in prostate cancer. We had a total of about 250,000 individuals included, and these participants are all from PAGE, the population architecture using genomics and epidemiology study. We did the analyses very similarly using fixed effect inverse variance, weighted meta-analyses with metal within each population and then across all populations. Here we're showing the multi-ancestry results. In total, we identified 43 significant regions, 4 of which were novel and 39 were known. So we wanted to develop next a polygenic risk score, and we actually did this using an independent study as the training data set. So this study was just recently published in Nature Genetics, and it had 1.4 million individuals, so it's much larger than our sample size, and it had a pretty substantial amount of diversity included. So we used the results from this study to develop our PRS. In total, they identified 582 variants that were significantly associated with type 2 diabetes, either in their trans ancestry analysis or within populations. And the first thing we did was to take these variants and look at the familial relative risk explained in our populations. And you can see that the familial relative risk explained ranged from about 12% in African ancestry individuals up to about 15% in Hispanic and European ancestry individuals. So just right off the bat, you can see the familial relative risk explained for type 2 diabetes is much smaller than it was for prostate cancer. And knowing that type 2 diabetes does have a strong environmental component, we don't necessarily expect that the PRS will do as well as it does in prostate cancer. And indeed, we see that to be true. So this is showing the primary results after applying the PRS to our populations here. You can see that the odds of type 2 diabetes in European ancestry individuals is about 3 for those in the top PRS decile. It's pretty similar actually in our East Asian population, and this is really promising. It's probably because there's such a large East Asian population in the discovery data set. It's lower in our native Hawaiian population, a little bit lower also in our Hispanic population, and lowest in the African ancestry population. And this is actually a little bit disheartening because they do have a pretty sizable number of African ancestry individuals, but I think maybe just for type 2 diabetes, we need maybe much larger sample sizes because it's not quite as heritable in order for this to do better. It could also be related again to the heterogeneity that people have mentioned earlier today. Maybe that's why we aren't quite able to capture variances as well in African ancestry populations. But I just want to highlight that here, we actually look at 2 different PRSs for each population. We look at the multi-ancestry PRS, which is represented by the diamond, and we look at population-specific PRS. So the same variants are included in each, but we use different weighting schemes with the weights being from the particular population. And you can see that we typically see that the multi-ancestry PRS has a slightly higher odds ratio across populations. This isn't always true, but it's typically true. And then when we look at AUCs, again, we see that AUCs are lowest in the African ancestry population, and they're highest in the East Asian and European ancestry populations. And comparing the different weighting schemes, the multi-ancestry weight typically, well, always has either a higher or identical AUC compared to the population specific. So this is really just reiterating that the multi-ancestry PRS does better than the ancestry specific. And this is consistent with what previous studies have found, that multi-ancestry weights do better. And there's a couple of reasons for this. One is obviously multi-ancestry is based on a much larger sample size, so the weights are just more likely to be accurate. But the other is that multi-ancestry weights are more likely to be able to converge to the true causal effect of a variant than we'd be able to do in just one population. So the last thing that we did in this study was to stratify the sample by age again. But here, when we stratified by age, we didn't see significant differences in predictive ability. But we also stratified by BMI, since that's a relevant question for type 2 diabetes. And we do see significant differences when we stratify by BMI in predictive ability, such that individuals with lower BMI have larger odds ratios, have increased risk for prostate cancer, or sorry, type 2 diabetes based on their polygenic risk score. And then when the polygenic risk score doesn't do quite as well in individuals with high BMI. And then we see particularly significant differences in the top 2 deciles. So this makes sense. This is suggesting that those with lower BMI, their type 2 diabetes onset was more likely to have a genetic component, whereas those with higher BMI are more likely to have an environmental component, which makes sense. And then overall, just to kind of summarize the type 2 diabetes work, we'd see that it doesn't do quite as well across populations as we observe for prostate cancer. I think there's a number of reasons that this could be, but I think one of them is probably just related to the environmental component for type 2 diabetes that we're not really able to capture using genetic information. And we probably need much larger sample sizes to do better in type 2 diabetes because it's not as heritable as prostate cancer. So to kind of summarize what I've shown today, I hope one of the biggest takeaways is that it is crucial to include diverse populations in genetic investigations. This can improve risk models across populations, and as we've shown, and also uncover novel biology that we wouldn't be able to do using a single population. We performed the largest and most diverse prostate cancer GWAS to date, which led to identification of 86 novel variants, bringing the total to 269. We developed a PRS that is highly predictive of prostate cancer risk across populations. We found that about 50% of aggressive cases fell in the top 80 to 100% PRS category, and that the PRS modified risk conveyed by pathogenic variants on prostate cancer risk. And then I didn't talk about this part, but I'm happy to talk about it if anyone has questions about using metabolomics to try to understand genetic risk of prostate cancer. And we also see that the effectiveness and utility of PRS is highly trait dependent. So when we look at the type 2 diabetes PRS, it doesn't do quite as well across populations as we saw in prostate cancer. And multi-ancestry PRS methods are still very much underway, but like as we've shown, I think PRS performance is really highly impacted by the sample size of the discovery population, the heritability of a given trait, and the genetic architecture of that trait. And really, like, I think it's great that we're working on a lot of these different multi-ancestry PRS methods, and I think there's a lot of potential to improve the predictive ability using these methods. But increasing the number of non-European ancestry individuals included will be probably the most effective way to improve the predictive ability of polygenic risk scores. So the work that I presented today was really on the behalf of many people who have been involved in these studies for a very long time, but I'd especially like to thank my mentors, David Conti and Chris Hyman at USC, all the investigators in the Practical Consortium, which includes many more people that I could fit on a slide, the investigators of the AAPC Consortium, and of course my sources of funding, which include an NCI K99, the ARCS Foundation, and the many other studies, many other grants that have contributed to these studies over the years. And thank you all for your attention. Sorry that I went a little bit long, but I'm happy to take any questions if we have time.
Perfect. Let's open it up for questions. We do have time.
Hi, Burcu. Nice to
Hi,
see you.
Prav. Nice to see you too.
Yeah. I was wondering, the 4 categories for ancestry that you have, are those based on genetic information that you guys categorize yourself, or is that self-identified?
So it's a little bit of both. Initially, it's based on self-identified ancestry, but we do a principal component analysis to see if there are any outliers from those populations, incorporating populations from 1,000 genomes to try to better categorize participants. So we exclude any individuals who are extreme outliers based on that principal component analysis, with the exception of Hispanic men. And that's probably true for Paige as well, because they're such a diverse population that we can't really define them by a particular category. So for Hispanic, we include everyone. But I do think this is like a huge limitation. First off, that we exclude individuals at all based on ancestry, and that we are kind of crudely categorizing individuals because there is so much admixture within any given population. And I think in the future, I'm hoping that the field will really move towards using these like mixed models instead that incorporate genetic ancestry a little bit better, so that you don't really have to categorize individuals by a particular population.
And the ones that they did self-identify, was that an ethnicity question, or was that a specifically ancestry question?
Yeah, it was. So there are many studies that contributed to this,
I see.
like a lot. So it's probably different for every study, but I would guess that it's usually a question that's more like race, ethnicity than ancestry. Okay. Yeah.
So that's kind of the lead up to my main question, which is I don't know if there is a way to even include race as a confounder, especially, at least we know in the US, race and racism is a contributor to a lot of the conditions that you were talking about. So I'm wondering, is there a way to use that as a confounder? Because that's also kind of what you're measuring with the genetic data.
So I do think that is like the way to go. So there's a recent publication by the Page study that was published in Nature, where they do this pooled analysis using mixed models instead of doing the fixed analysis or the meta-analysis across populations. So when you do the pooled analysis, you include like a genetic relationship matrix, and you don't exclude individuals or categorize them by their ancestry. But another thing that they did was to include a variable for like self-reported ancestry, which could be self-reported race, ethnicity, depending on the study. And that is like the purpose of that is not really to capture genetic differences, but more so like potential, just maybe social or environmental differences that could be experienced because of what population you identify with. So I think you could incorporate that. But, there are some problems with it. A lot of these big studies are collected across, like these consortia studies are collected across many smaller studies. So the questions are asked differently. So there will be heterogeneity in that question alone. But that's at least one way that it could be done.
Okay, thank you.
Mm-hmm.
Hi, Burcu. Thank you for the very comprehensive talk. And so I have maybe two questions. The first is a quick one, which is that you mentioned the contribution of both common and rare variants. I wonder if you actually see evidence for interaction. So the rare, potentially more penetrant variants showing very different effect sizes for people with high and low polygenic risk, or are you basically saying that both common and rare variants strongly contribute maybe to the disease risk in a complementary fashion? So that's the first question. The second question is, I've never worked on cancer genetics myself, but I've always been very curious because obviously another side of the story is somatic mutation. But then the biology there is very, very well understood, right? You have the oncogenes, you have the tumor suppressor genes, but then in GWAS, do you see the similar genes showing up at all?
Yeah.
And if not, what is the GWAS doing, right? So then is this suggesting a completely different kind of pathway, or if, for example, the polygenic risk score actually associated with somatic mutation loading? So I guess that's sort of a general question about the relationship between germline and somatic mutations in cancer biology.
Yeah. So for the first one, we did test interactions. So I only showed like the aggregate results looking at those 4 genes in aggregate, but we did look at the 4 genes separately also, and we do see similar relationships as shown for the aggregate. But in terms of testing interactions, the only place that we saw a significant interaction between rare variants and the PRS was for HOXP13. And I think that's because HOXP13 has such a large effect. Like of all the variants of all the individual genes we look at, HOXP13 definitely has the strongest effect on prostate cancer. So that's just probably where we're best powered to be able to see a significant interaction between carrier status for rare variants and the polygenic risk score. And then the other question about, so we do see some similar genes that show up in GWAS studies as we identify it in somatic tissue. I haven't really done a lot of work with somatic tissue, so this is just kind of a little bit of what I know. But I think BRCA2 is something that shows up in somatic tissue for risk of prostate cancer. And it's also something that comes up in our GWAS studies. And when we look at rare variants in BRCA2 using sequencing studies, we do see that there's an association between these variants and risk of aggressive versus non-aggressive prostate cancer. But in terms of like, I think what you mentioned about looking at like somatic mutation load by PRS, that's a really interesting question. I don't know if it's been done, but that would be a really interesting thing to do. I know that there has been some work recently, maybe not necessarily in prostate cancer, but in general, looking to see if there's some relationship between germline mutations and somatic mutations. And I think there has been a little bit that's been found, but I'm not too familiar with literature to know much about that. But I think that would be an interesting question to see if PRS, like from germline PRS, is associated with mutation burden in somatic tissue.
But is it fair to say that if you do some sort of pathway enrichment analysis for the top hits you identify in GWAS, you do not really see tumor suppressor showing up as the obvious answer?
I don't know. When we do a pathway analysis, I think the types of things that show up are related to like inflammation. And that could also be related to like what you see in the tumor as well. But in terms of like enrichment for a particular gene, I don't know if there are like any genes that show up enough to show it that one gene is really enriched, Yeah. But I think for prostate cancer, I'm sure that the tumor is involved in risk, and especially involved in risk when it comes to like whether or not the disease is aggressive. But germline genetics has really been like one of the strongest risk factors for prostate cancer.
Yeah, that's fair. Yeah, thank you.
Yeah, so I just want to say again, this has been a really fascinating presentation. I probably understood maybe 15–20% of it.
Oh, I'm sorry.
I don't think that's because of your, I think that's more on my end. I'm just going to take this unique opportunity to, I'm going to take this unique opportunity to ask a question of an expert in the field. So I've heard that the BRCA gene that's been identified for breast cancer, when present in men, also predicts prostate cancer. Have you heard anything about this, or what are your thoughts on that?
Yeah, so there are 2 BRCA genes. There's BRCA2 and BRCA1. Those are both like really big risk factors for breast cancer. BRCA1 is actually not a very big risk factor for prostate cancer. I think it's considered to be one, but the odds ratio is pretty weak. BRCA2 is actually a pretty big risk factor, but not as big a risk factor for overall prostate cancer, but a pretty big risk factor for aggressive prostate cancer. So like we did a whole exome sequencing study recently in like 5,500 men with aggressive and non-aggressive prostate cancer, because I've been showing a lot of this work looking at the polygenic risk score, but that's really based on common variants. But we don't see that common variants are really associated with aggressive disease. So we kind of suspect that maybe rare variants are more important. And when you're looking at BRCA1 and BRCA2, that's really typically focused on like rare pathogenic variants within those genes that are impacting risk. So rare pathogenic variants in BRCA2 are associated, like in aggregate, just being a carrier for any of those variants in BRCA2 is associated with like I think about a 2-fold increased risk of aggressive as opposed to non-aggressive disease. So it definitely is. It's not as strong a risk factor as it is in breast cancer. Like in breast cancer, I think the odds ratios are much bigger, but it's certainly a risk factor for aggressive disease. And I think that's true. Like, you know, these are both hormonal diseases. So I think there's a lot of overlap in the genes we see, like CHEK2 and ATM and maybe POLB2 are also risk factors for both breast and prostate cancer. There's a lot of overlap, but the effects are usually a little bit different across the diseases.
Hi, Burcu. I have a question. I want to just echo what my colleagues have said and say that this is really important and super fascinating work. I can see how the work that you've shown today will hopefully spur a lot of other fields to do the same thing. Of all the things that you presented, I think one of the things that was most interesting to me was your finding that, and this is more of a clarification, but I also have a question about it, which was showing that those who had the lowest polygenic scores for breast cancer also had the highest preponderance of carrying the HOXB13 rare variant allele, right? So if I have that right, I'm not quite familiar with any mode of inheritance that would explain why there would be people with lower risk scores who have these, and these are common genetic variants in the PRS. So why would they have higher risk of carrying this particular allele relative to the other risk categories? Because I would have assumed that every category has an equal risk of carrying the particular allele. Could you explain that or flesh that out a little bit more?
Yeah, I mean, it's possible that there could be some selection bias somehow playing into it. You know, we have to identify these cases, and maybe when we identify cases, maybe they're just, I don't know. So I think part of it is that HOXB13 is very rare, and you know it's also somewhat rare to have a high polygenic risk score, and it's just unlikely for both of these things to happen together. So I think it's kind of just that there are like these two independent pathways that could be leading to prostate cancer risk. If you carry HOXB13, that like might be enough on its own to develop prostate cancer. If you have high genetic risk, that might on its own be enough to develop prostate cancer. If you have both of them, you're probably very likely to develop prostate cancer. But it just seems that it's probably pretty rare for them to happen together because HOXB13 is so rare to begin with. So I don't really think that there's like a true biological relationship between the two. I think it's just kind of something that like tends to happen, but like I can't imagine why like having a high PRS isn't going to make you more or less likely to have a particular variant biologically. But I think having prostate cancer means that you're trying to understand, why did this person develop prostate cancer? If they didn't have a high PRS, maybe it's because they carried a rare variant. I don't know. Does that make sense? It's kind of how I've been thinking of it, but it's a little bit, yeah. Yeah.
Is it reasonable to also make the interpretation that people with the low PRS are protected from?
Yeah, definitely. I mean, I think that's kind of what we'd hope to see. So, but you know, I think in terms of like, yeah, I think they definitely seem to be protected against prostate cancer when they have a low PRS. The odds ratios are like well below 1. But you know, in terms of like trying to maximize power to detect rare variants. If you have this question of what are the rare variants associated with prostate cancer, I think if we got a bunch of cases that had low polygenic risk scores, that would be kind of an interesting question, because you would expect these cases with low polygenic risk to be less likely to develop prostate cancer, but for some reason they still did. And maybe those are the men where we could really maximize our power to detect rare variants, because they're just potentially more likely to have rare variants.
Thanks.
I had I think a similar question to James, and I wondered, I think I'd phrase it in some sense generic question about case control studies like this. And I think this is where you were going just with your answer, which is that it seems like you'd have to do a lot. You love to do a lot of thoughtful analysis in creating the controls, the people, because I guess my point, the obvious point is that some set of the controls will be cases in the future, and you'd love to know which ones those are, right? What would you say? What's the answer to that as currently practiced? When you have all these limitations, you have all these cohorts you're jamming together, and they each have their own recruitment, so on and so forth. What's the off-the-shelf defenses against this problem?
So I think one of the best things you could do would be probably to choose controls that were older in age. Of course, then you can't really address questions about age, but if they're older in age, then they're less likely to, and then they haven't developed prostate cancer yet, then they might be more likely to be a true control. And if they do develop prostate cancer at the age 80, it maybe was not really very genetic in origin if they developed it at a much older age. So there's still probably a better control than at least somebody who developed prostate cancer at the age of 50, which would be probably much more genetic in origin. So that's one thing. I mean, but it's hard to do across all of these like different studies. But I think one thing that's really good, like in the multi-ethnic cohort, we have, there's like 200,000 individuals were initially enrolled in the multi-ethnic cohort in the 90s. And every few years, those participants are linked up to SEER registries and like also California and Hawaii cancer registries and death registries to figure out what their status was. So if they initially were control and became a case, we would know that with the multi-ethnic cohort. Of course, that is not going to be true across all studies, because it's a lot of like money and time and resources to continually link up studies or participants to registries. But if we had the resources, that would be a good way to do it, to make sure that somebody remained a control throughout their life. But also with prostate cancer, it's tricky because at autopsy, I think a ridiculously high percentage of men turn out to be prostate cancer cases, but they're not aggressive cases. So they shouldn't be diagnosed because they shouldn't be treated, because it wouldn't have changed their life course, So it's really tricky with prostate cancer, because as you get older, it's just so common to have prostate cancer. And the real question is it going to be aggressive prostate cancer or not?
So I'll step in and fill a little bit of the quiet here, just with another sort of general question. So my understanding is that we are genotyping cancers. And so are we able to identify the more aggressive prostate cancer genotypes from the other ones? And to what extent do your models change when accounting for these different types of prostate cancers?
Yeah. So I think, so we haven't really been able, so I did mention earlier that particular 8q24 variant that is associated with aggressive disease. That's kind of a unique example, but outside of that, typically if we do a GWAS on like overall prostate cancer, and then we do a GWAS on comparing controls to aggressive prostate cancer, the variants that we identify are the same as what we identify for overall prostate cancer. And the effects that we see are actually about the same. We don't really see that like there's a stronger association with aggressive prostate cancer. And I think that's just because just this limitation of looking at common variants. Another limitation is maybe the sample size for aggressive prostate cancer cases is not as big as it would need to be to identify these variants. We have a much, much larger sample size of overall prostate cancer than we do aggressive prostate cancer, because aggressive prostate cancer is not nearly as common. But we typically identify the same variants if we're even powered to identify them. But when we look at the overall prostate cancer variants and see if they're associated with aggressive disease, we see about the same odds ratios show up. So I think like a good place to look for variants that are specifically associated with aggressive disease is with exome or whole genome sequencing data where we can look at rare variants. I think rare variants will likely be the place where we can figure out if there is like what the genetic contribution to aggressive disease is.
So just so that I'm clear, when your use of the term variant in that in your answer, you're speaking to the sort of risk side of the equation, sort of the right side. I guess what I'm asking is, so to drill down, when you say, how do your studies determine between aggressive and non-aggressive? Is that sort of like the doctor says, okay, this was aggressive? Or is it that you actually have genetic data on the kind of cancer that the person had, and then you're parsing, you're discriminating against across different kinds of cancers with genotyping of the actual cancers?
Yeah, so it is determined from like a physician standpoint. They do like staging of the disease. So you can, when you do biopsy of like the tumor, there are certain like features that you'll look for in that biopsy to determine how aggressive it was. But there aren't really, for prostate cancer, I don't know, like when we look at somatic tissue in prostate cancer, I don't think that there's a clear signature in terms of genetics that would define aggressive versus non-aggressive. I don't know, that it's possible that there is, and I'm just not as familiar with the somatic literature, but when we define aggressive prostate cancer in our studies, it's based on what the physician has said in terms of like the grade and stage of the disease. And those things get reported back to like the SEER registry. And we link that up with our data sets.
Any other questions? I was going to have Burcu lead us out with a question of like stepping back and maybe lessons learned on, if people in the audience have other outcomes in mind that aren't prostate cancer, but are really interested in having diverse cohorts and doing the type of work you're doing. Do you have thoughts on those type of lessons learned that you'd like to share?
Yeah, I mean, I think, a lot of what we see is in prostate cancer, we see a huge improvement in polygenic risk scores by including like not that many non-European ancestry individuals. I just think that there's so much potential to really improve our risk models by including a much more representative like number of non-European ancestry individuals. I think that's like definitely the biggest takeaway I hope that people get is that, and really one of the reasons is that the LD structures in different populations are very different. And if we just limit it to European ancestry populations, we're not going to be able to really accurately identify causal variants. But if we include diverse populations that have different LD structures, we'll be able to get a little bit closer to the true causal variant, and that will improve polygenic models for all populations. Other thing that like kind of moving along this line of causal variants, another maybe limitation of this type 2 diabetes PRS that I showed. So the study that we used to develop this, they didn't do fine mapping. And I think that, maybe because we're not really capturing the causal variant as well, that could be part of why the PRS doesn't do as well across populations. But, I think if you want to do a good, like as good as you can for a polygenic risk score, first off is include as many non-European ancestry populations as you can. Fine mapping across the different populations will get you a little bit closer to the true causal variant. And I think that could really improve the predictive ability. I think that's part of why we see that our PRS of 269 variants, which we did using fine mapping and the multi-ancestry weighting, I think that approach works maybe better than some of the genome-wide approaches because of the fine mapping we did. I think we just kind of got it as good as we possibly could for now, at least with what we know. But those are some of the, like, I don't know, some of the thoughts that I have. But, you know, this is also a continuously changing and evolving field. And what's good today might not be as good tomorrow as we learn new things. And what works well in one trait might not work well in another trait because the genetic architecture differs between traits and the heritability differs between traits.
That's fabulous. I'd like everyone to join me in thanking Burcu for joining us today.
Thank you.
Thank you. That was great. Thank you.
Yeah, thank you for having me.
And thanks everyone for joining our working group meeting. And we'll see you all next week.